So this is a really quick overview of cellular respiration. We went into a lot more detail in the lecture on this topic, but I just want to give you enough to refresh your memory so that you can understand what we're going to do in this laboratory activity. So first of all, recall that the mitochondria is really important in cellular respiration. We refer to that organelle as the cell's powerhouse. So that's what this drawing is right here. This is showing us that two parts of cellular respiration, the Krebs cycle and the electron transport chain, take place in the mitochondria. The third part, glycolysis, um, does not take place in the mitochondria. It takes place in the cytoplasm. So remember, the big picture here is that we are using oxygen to break down molecules in our food, like this glucose molecule. So in glycolysis, we start with this 6-carbon glucose molecule. It gets cut in half, the splitting of the sugar. We end up with 2 3-carbon molecules at the end. We make a little ATP and we load up some high energy electrons in this NADH molecule. These 3-carbon molecules are going to be modified slightly and then they're going to enter the mitochondria where they're going to be further broken down in the Krebs cycle. So as you can see here, the rest of this carbon is being emitted as CO2, which is a waste product. So the inputs for cellular respiration are this glucose and oxygen. We'll get to the oxygen in a minute, so that's the oxidizer. CO2 is one of these waste products. Water is another waste product, and we'll get to that in just a second too in this overall equation. So the Krebs cycle basically breaks down our food to CO2, this waste gas that's released when we exhale. But the other thing that's happening in the Krebs cycle is we're loading up more of these high energy electrons onto the taxis, the NADH and the FADH. So all of these high energy electrons then go to the electron transport chain in the inner mitochondrial membrane. Remember, the mitochondria has these two membranes. So basically what happens here is the energy, you can see all these NADHs and the FADH are coming here. They're dropping off the electrons. That energy in those electrons is used to generate a proton gradient. The protons go back to the other side down their concentration gradient through this amazing protein ATP synthase that we learned a lot about that adds a phosphate group to ADP forming ATP. So that's where the ATP comes from. We're breaking down food like glucose, these molecules in our food, and we're converting the energy to the ATP, which is the form of energy that our cells require for so much of the work that they do. So this electron transport chain is also where the oxygen is needed. It is pulling the electrons through the chain. So we can see the oxygen here in this diagram. Once the oxygen gets those electrons, it gets converted to water. So that's where that waste product is generated. So in this laboratory, we are going to be measuring cellular respiration by measuring the consumption of oxygen. So oxygen and glucose are required to make this happen. And as cellular respiration proceeds, oxygen gets consumed. When you inhale, you are consuming oxygen out of the atmosphere. When you exhale, you're getting rid of CO2, which is a waste product of cellular respiration. Okay, I hope this helps, and I hope it helps you understand the activity that you're going to look at using a respirometer, which examines the consumption of oxygen and uses that to measure the respiration rate.